Hello and welcome to the Beyond Biotech podcast number 99. I'm Jim Cornell and this is the weekly podcast from Labiotech. This week on the podcast we have a conversation about treatments of type 1 diabetes. Our guest is Anders Essen-Möller, founder and chairman of Diamyd Medical, which is addressing type 1 diabetes and latent autoimmune diabetes in adults through its product Diamyd. This is an antigen-specific immunomodulatory therapeutic for the preservation of endogenous insulin production. All right, so I guess the first and hopefully the easiest question is if you could give me a little bit of background on the company and how it was formed.
Okay, yes. So it's a pleasure to be here. I'm Anders Essen-Möller. I'm the chairman and the founder of this company, Diamyd Medical, who has come rather far in developing a pharmaceutical for type 1 diabetes. It really started in the 90s when my daughter, 10 years old, came out with type 1 diabetes. It was a dramatic incident, and I had some experience from the disease. I worked with on the medical technology side before, on hemodialysis and peritoneal dialysis and also on metabolic diseases. I came from the medical technical background, and I had started a medtech company before, software company, when my daughter came out with type 1 diabetes. So what happened was that I traveled around trying to find something that could help my daughter. And I met Åke Landmark in Kyoto in Japan. And he tipped me about this molecule GAD65. And I looked up the GAD65 and I found that there were basically 3 universities working with it. There was UCLA with Dan Kaufman, and he had just published something about GAD65. So I went there and licensed the rights to develop the product in 1994, I think. And then I went to University of Washington and licensed their intellectual property rights. And I went to University of Florida and met Mark Atkinson, who is now a professor in diabetes and also on our board of directors. And I licensed his intellectual rights. So from having 3 antagonists, the University of Washington, Seattle, and UCLA, there were now 3 friendly universities that all wanted 3 American technologists or professors that wanted the same thing. And so we had suddenly a very broad expert team that could help us design, what to do. And we met in Sweden later on, a couple of months later, and we had this guy, Hans Wicksell, who at that time was the rector of the Karolinska Institute and professor in immunology. And he came in, he was very busy, he came in and said, well, do like this, take the drug, take the whole molecule, don't try and find parts of the molecule, let the immune system decide which parts of the molecule it should react against, and formulate it in all of them. Try 5 doses, 4, 20, 100, 500 micrograms, do a boost, a prime and a boost, and then he went. So that's what we do. We know that type 1 diabetes is an autoimmune disease where the immune system attacks certain parts of the beta cells. It could be insulin, it could be GAD. There are basically 2 main lines of the disease. One is a insulin-induced diabetes and the other one is a GAD-induced diabetes. That is based basically on genetics. And why the immune system reacts against these beta antigens, autoantigens, we don't know for sure. When it comes to, for instance, GAD, which is produced in the beta cells, it could be that the patient has had a previous disease, with a virus that is similar to the GAD antigen, or it could be that there is something wrong with the beta cells. It could be that the GAD antigen hasn't been seen by the immune system. It could be that it has been a sequestered antigen, that suddenly when it gets disposed and you have the right gene to sort of detect it and present it in a bad way, then you could develop autoimmunity against then GAD65. When it comes to insulin, it could be similar things. It could be that you had an infection when you were young. There are protocols where people try and vaccinate young guys to see if they can prevent diabetes that way when they are real young. It could be something wrong with how the insulin molecule is developed in the beta cells, proinsulin, etc. So there are many, many ways how type 1 diabetes could develop. Stress, different habits, genetics, infection, etc., etc. So we were happy to find this solution already then. I mean, in the 1995, we knew that if we could stop the autoimmunity, if we could just stop that, then the beta cells would not deteriorate. So what happens in the immune system when the immunity is going on is that first it starts attacking the beta cells. Then the patient may experience high blood sugar without knowing it. And then after a time, there are so few beta cells left, or the insulin production is so low that the patient ends up at the hospital and he needs to take immediately insulin injections for the rest of his life. If he doesn't do that, he dies. So it's a good disease to treat. I mean, you could save the life, so that's nice. But of course, it's terrible for the patient. He needs to check his blood sugar every day. He needs to take insulin every day. His mother and father checks on their phones what their kids have as blood sugar levels in school, and they call and they don't know if they should call the guys and ask them to take extra insulin injections. So it's psychologically, it's a terrible disease. It's really bad. It sort of screws up the life for many, many people. So there is definitely a big need to prevent the disease and to cure disease. Preventing the disease is going on. We have now a drug that is approved by FDA, where an anti-CD3, it's an antibody against anti-CD3, an anti-CD3 antibody that sort of suppresses the immune system. It sort of eliminates the T cells, and the T cells are those attacking the beta cells. So they get destroyed, and in that way you can delay the disease for about 3 years at the moment, which is very good. It's just that if you don't have T cells, you can get a lot of other immune diseases. You may get real bad diseases. I won't even mention the names. I mean, there are terrible diseases you can get if you don't have, if you suppress it, if you take out your immune system. And moreover, what is good for us in a way is that they managed to get a very high price, like $200,000 for a 14-day treatment, where they get these injections of anti-CD3 antibodies against the immune system. And that is, of course, expensive. And it paves the way for other therapies like ours that don't sort of compromise the immune system. We go direct to a certain antigen. With that, I stepped back, and when we started to develop this medicine, we started with small studies in LADA patients and in type 1 diabetes patients. There are about 500,000 new type 1 diabetes in the world every year, and 50,000 of them have the gene that we treat at the moment. In the beginning, we sort of treated everybody. We didn't know about the genes. And we got as far as to phase 3 studies. We teamed up with J&J. They paid us a lot of money, and with milestones. It was one of the biggest deals in Sweden, one of the biggest biotech deals in Sweden. We did 2 phase 3 studies, one in Europe and one in the States. And when we looked at the results from the European phase 3 studies, we saw it worked, but didn't work well enough. So we wondered, why doesn't it work well enough? And we couldn't come out with the conclusion. The COVID, could it be? No, that was not COVID. That was sort of the swine flu, different types of flus. Could that interfere? We didn't know. And we looked at some of the old studies. They worked. So we took the money we had in the company, like several 100 million crowns, and gave that out to the shareholders. And we kept 50 million Swedish crowns, and we kept the data and the names and etc. And we started all over, and we made a small new financing and started out trying different. It was like a living AI brain. I mean, you try, does this work? Does that work? And then you learn and you come up with some new ideas. Does this work? And you try it and you learn and you get the results. And what we did, we looked at the genes, for example, and we found that there were certain genes that where our drug worked. There was a haplotype, DQ2 type gene, where it seemed that GAD worked. And there was a haplotype DR4 DQ8 gene, where it seemed that the insulin antigen would work. And that was about half-half, 55% of the Western population at least have this DR3 DQ2 gene. And we did new studies where we found, and meta-studies, where we I think we had like 15 studies where we looked, does it really work? Yes, it really does work. And we filed patents, of course, of that, and we have good IP protection for the GAD autoantigen and DR3 DQ2, and we have some IP protection for the DR4 DQ8 with the insulin antigen. So that looks very good, and it looks real. We published our meta studies, and at the same time there were big Batalagia published their big studies where they also found that there was a GAD form of the type 1 diabetes and there was an insulin form of type 1 diabetes. So it was really a discovery. I mean, it was discovered. It was like the brains, the AI brains, they found this and they found that and they checked it and they came up with results. And now you need to sort of just confirm what you have learned. And we are doing that right now. We are doing a phase 3 study, 300 patients, 8 European and US, where we inject GAD in those that have that particular gene. And there was one more discovery made, and that was in allergy. You had pet allergy and wheat allergy, grass and pollen and stuff like that. Usually you get, when you get allergen treatment, you inject the allergen, and you could do that for years. I mean, weekly or monthly injections of allergen, and it works, but it takes time, and it's a real cumbersome for the patients to go through that kind of therapy. And then there were guys who tried to inject the allergen directly into the lymph system, the lymph nodes. They found that they could, with only 3 injections, more or less cure the allergy long-term for years. Instead of treating for years, they could do 3 injections once per month. And they could also use much less allergen. I mean, they could use 100th of the allergen that they used in these long-term allergy treatments. So we tried, Johnny Ludvigsson, he said to us, try this directly intralymphatic. I mean, we did some inventions there. And so we decided to try 4 micrograms, that's almost nothing, 3 times directly in the lymph node. So we started safety study, Diamyd 1, and then we went further with Diamyd 2, 2A study, 2B study, and we found that it worked again in those DR3 DQ2. So, I mean, it's really that we know it works in the DR3 DQ2. There are other genes. There may be genes where it doesn't work. There may be genes that helps, etc. But it works in DO3DQ2. So it's really a matter of just doing it, and we are just doing it. We have now 110 patients in, and there is a lot of discussions. What is really a treatment of type 1 diabetes? We say that we are treating the beta cells. The beta cells are being destroyed by the autoimmune system, so we try and save them, saving beta cell function. And we can do that by measuring how much insulin they produce. I mean, if they are attacked, they don't produce insulin. If they are not attacked, they produce insulin. If they are left. So we need to save them from becoming destroyed and save them from being attacked. And by doing so, we treat the type 1 diabetes. There are those who say that we need to show that it also improves the long-term A1C levels, the glucose levels, the blood glucose-related levels. And of course, that's good. But I mean, there are so many ways where you can keep your blood sugar levels in check. You can eat good food, you can go out and walk, you can work out, you can take different pills. If you, in spite of that, has too high levels of blood glucose, you take more insulin. So, I mean, really, the doctors, they are there to treat your diabetes, to keep your blood sugar level in check. They don't bother about how much insulin you produce yourself. So they don't really bother about the disease, the origin of the disease. The origin of the disease is how much insulin can you produce insulin. If you can produce enough insulin, you don't need to do all these things. You can eat sugar. You don't have to eat gymnast to work out, etc. Of course, it's good to do that too. And now with the new GLP antagonists, etc., you sort of reduce your body fat and you increase your insulin sensitivity, which is good. All those things are good. So that's a parallel thing. But still, what you want to do, you want to have your insulin cells to produce insulin. And you know that if you can do that, you have the foundation of the new pharmaceutical industry worldwide. There are about 500 million with diabetes, of which 10% at least is type 1 diabetes patients, and you have an autoimmune form of the type 2 diabetes, the LADA. So, I mean, it's a huge, huge multi-billion industry if you can sort of prevent the beta cells from becoming distracted. And then, of course, you have to make sure that they increase their insulin producing capacity. The best thing is, of course, if you can stop the beginning of the disease once beta cells are becoming attacked and the disease is progressing towards insulin dependency. If you can stop it before patients become insulin dependent, then you have hit the goal. And there is no reason why we shouldn't be able to do that. I mean, it's really, if it works when you have type 1 diabetes, it works before you have the type diabetes because it attacks the same molecule. It's the same molecule treating the same cells. So I would say it's a done deal. It's just a wait. We say we'll stop the diabetes in its tracks, and that's a good thing.
Are there different approaches to tackling type 1 diabetes other than the treatment that you're using?
Yes, there are many. I mean, over the past 30 years, I think there have been hundreds. And I mean, you have the anti-CD3 antibodies, which is now approved in the States to prevent open type 1 diabetes, which is priced at $200,000. You have anti-CD20 antibodies, which is against the B cells, B lymphocytes. You have different types of antibodies, IL-21, IL-17, etc., trying different sort of things, changing the immune system. You have ATG, antibodies to rabbit. You have new humanized antibodies similar to the ATGs. You have GLP-1 antagonists that sort of works for type 2 diabetes and for fat, and they also induce better insulin production, etc., etc. So there are so many, and you have the stem cells coming. If you have stem cells from your relatives, you don't need immune suppression, but if you get stem cells from somebody else, you need lifelong immune suppression. If you encapsulate your stem cells, you may have to exchange them, etc., etc. So really what it comes down to is to treat the disease, and that is to keep your beta cells from doing what they're supposed to do.
And I guess the number of cases of diabetes seems to be on the rise. How important is it to, because it's a global issue, how important is it to try and keep costs down?
Yes, it's very, very important. I think in the States, the cost for type 1 diabetes is more than $100 billion per year. And there are those who have the hypothesis of cleanliness. I mean, the more clean you are, the more you wash, the more antibiotics you take, the less bacteria you have in your bowel, etc., with the microbiomes, etc., the more autoimmune disease may come up. I mean, the immune system is there for a reason. It has to be constantly working to prevent cancers and all sorts of diseases, to take control of your body. So the more medicines out there, the more diseases you're probably going to see, and the more expensive it's going to be.
Are you hopeful in the long term that type 1 diabetes can be at least managed and maybe even cured?
Yes, I'm very hopeful. As a matter of fact, I'm convinced that it's going to be cured. I mean, with antigen-specific precision medicine, you'll be able to sort of stop the autoimmunity. And then if you do it already before type 1, the stage 3 has broken out, then you're on the way to a cure perhaps by regeneration. The beta cells that are there, they may regenerate, or you may stimulate regeneration with additional therapies.
Type 1 diabetes is definitely a condition that most people are aware of, and with the numbers of patients rising, it's great that companies like Diamyd are confident that they're able to make a positive difference. Don't forget to check out the latest news and articles over at Labiotech.eu, and I hope wherever in the world you are, you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Beyond Biotech.